Yeah, good morning, everyone. My name is Fabian Müller, and it's my great pleasure to be hosting this last edition of the SCOG virtual lecture series before the summer break today. Today we'll have Carsten Marr from the Helmholtz in Munich with us. But first, as usual, we go through a couple of housekeeping announcements. So, as I said, we will have a little summer break and then resume the lecture series in September 10th, with Peter Tessarts from Cologne. And now I would like to introduce Carsten. So Carsten is the deputy head of the Institute for Computational Biology and the group leader at the Helmholtz Zentrum in Munich. So he did his diploma in general physics in Munich as well, and then did his PhD at the TU Darmstadt, after which he then went for a postdoc at Jakobs University in Bremen, and then shortly thereafter he joined the Helmholtz Zentrum for the first time in 2008 as a postdoc. And then in 2011, he went for another research stay at the University of Edinburgh on a DFG fellowship. And in 2013, he then rejoined the Institute for Computational Biology at the Helmholtz Zentrum. He has won numerous awards, among them an ERC Consolidator Grant and the Erwin Schrödinger Prize, as well as the CSB2 Prize in Systems Biology. So he's very much interested in gene regulation and networks and the modeling thereof in pluripotency and cell differentiation. And he basically was responsible for establishing the field of computational hematopathology. And I believe that this is going to be the focus of his talk today as well. So we look very much forward to the talk. So if you have questions, please raise your hand at the end of the talk, and we will basically call you up and you can ask the questions yourself, or you can post questions in the chat. And with that, I hand over to Carsten and we look forward to your talk. Thank you.
Fabian, thank you very much, and welcome everybody to this Friday morning lecture. I like this format a lot, and I hope you're also going to enjoy it. Let me try to share my screen here. So, Fabian, you see the slides?
Yes, we do.
Great. Welcome. I go right into it. By introducing what my group is doing. Basically, we are two-tiered. One part of the group is trying to boost image-based diagnostics with deep learning, which I'm going to show you in a second. The other part is trying to predict single-cell kinetics with mechanistic models, which I'm also going to show within the next 20-30 minutes. Let me start with the topic I'm going to look into now, hematopoiesis, the production of blood cells, although the group is also doing research on other systems. This is one of our prime interests, and I thought it fits nicely to the talk from Simon Haas 2 weeks back, who also shed light on a couple of aspects of this super important process. Why is it important? Well, we produce 500 billion blood cells every day. We need to do that, of course, because they have a high turnover, the amazing fact about that system here that it's so stable, right? 500 billion blood cells a day. And it's not just producing cells, but it's producing cells in the right amount. If you look at hematopoiesis in a bit of an abstract fashion, what we know is that we have these blood stem cells in our bone marrow, roughly a couple of hundred of thousands of them. They are not doing too much. Sometimes they divide and then they differentiate into progenitor cells, which are less potent. They are basically restricted to a couple of mature lineages. And then after a couple of division and maturation processes, these progenitor cells differentiate towards mature blood cells, which basically then do the job of e.g. transporting oxygen or fighting pathogens. So 500 billion of those every day. How is that done? I mean, there are a couple of super interesting questions, how that process is happening, how it is stabilized, what things go wrong in disease. And Simon told us a couple of super interesting facts about the niche, for example, where do the blood stem cells live in the bone marrow? What do they need in order to do their job? I'm going to look at a couple of other aspects, and one of the questions we quite early started to ask ourselves is, how do these cells actually decide, right? In this hierarchy here, you see this is there are a couple of decision-making processes in there. So blood stem cell has to decide to become either the MAGE progenitor or the GM progenitor, and then again these progenitors decide a couple of times in this hierarchical fashion before they become a mature blood cell. How is that happening? Why? How is it stabilized molecularly? So how do cells decide? In one very early proof of concept study, we used as one of the first labs, deep neural networks in order to predict blood stem cell fate. And this is a while back already now, this paper here. It was done together with Tim Schröder at that time, our collaborator at the Helmholtz Center, now at ETH. Tim took hematopoietic stem cell from mice, out of the mice on a glass slide, and imaged those cells over 1 to 2 weeks. And we had a lot of these time-lapse microscopy data around, and the question we asked here was, are we able to discriminate megakaryocyte erythrocyte progenitors, these MEPs here, from granulocyte monocyte progenitors, GMPs, just from their appearance in a bright field movie and basically the speed of the cells. So that was the task here. Are we able to discriminate those? We asked our expert hematologists in the lab, are you able to discriminate them? And say, well, are we looking at these images? Tough. So what we did was training a relatively simple at that time linear convolutional network with a couple of hundreds of thousands of those single image patches here, 27 by 27 pixel. So pushing that through a network, training convolutional filters and the parameters of that network, then adding the displacement, that is how much did these cells move between 2 time points. This gave us a patch linear score. So what does the network believe this patch belongs to, either the MEPs or the GMPs, and then averaging that over multiple timeframes of the life of a single cell, because we have a couple of minutes high resolution. This allowed us to now ask the network, what do we believe, is this cell an MEP or GMP? And we found actually that annotated cells, so those cells where we knew what they were because of a marker that was additionally expressed in the cells, we could actually predict quite nicely with an area under the curve, which is a precision or accuracy, or let's say a performance measure for the prediction, with an AUC above 80%. For those cells that were annotated. Maybe more interestingly, we could also predict the fate of the cells for so-called latent cells, that is cells where only 2, 3, or 1 generation later the marker turns on. So we can actually predict the fate of the cells up to 36 hours before the marker onset using our CNN. And we can also identify thus the cell fate without any molecular marker, so just on the bright field images and the movement of the cells. After that, we had basically a great tool to use morphology of single cells in order to predict their fate, and we were wondering, can we use that in a more clinically relevant setting? So we talked to a couple of hematologists, in particular with Carsten Spiekermann at the LMU. We started collaborating, and if you look at blood diagnostics in 2021, it's amazingly analog. So if you don't feel well, you go to the doctor. Doctor takes 1 drop of your blood, smears it out on a glass slide. This is then dried and stained, and then he goes to the lab. And in the lab, a couple of tests are done, but at the end, a morphological expert, a cytologist, will look at these blood smear and basically perform an algorithm, and that is select 1 to 200 leukocytes, white blood cells, randomly from the smear, and classify them. And based on the classification of the cells and based on the abundance of the different cell types, you'll be diagnosed into one of different diseases, right? So we wondered, of course, is that a process that we can automate using deep learning? Put differently, can we classify blood cells with AI? So that's a very general question. Of course, we zoomed in a bit here, and together with Carsten Spiekermann, we focused on acute myeloid leukemia, which is a very aggressive blood cancer, which requires immediate treatment. So we took and we wondered actually if we can identify blast cells, which are diagnostic for AML from benign leukocytes. So we went with Carsten to his archive. We retrieved 100 AML patients and 100 healthy controls. We scanned the blood smears of these 200 patients. We identified white blood cells in these scans, went back to the lab and asked the cytologists to annotate each of these 80,000 leukocytes into roughly 20 different classes. And then we asked a simple question, binary question, for the sake of simplicity: can we discriminate blast cells from benign leukocytes using, again, a convolutional network? In that case, it was a ResNet. And here you see again our area under the curve here, which is 99.9% here. So actually our trained neural network is able to discriminate these blast cells from the benign leukocytes very, very accurately, which is great. But of course, this number or this curve here doesn't tell you much, right? What you have to do is to compare it basically to the human annotator, to this algorithm the experts perform. So we went back to the lab and showed exactly the same tests that we had to another human cytologist or another rater, and asked her to perform the same task on the test set here twice. And this is actually what you get. The human expert did it twice with a 3-month delay between the two tests. And you see that there is variability, of course, in the human expert, and also, well, classification accuracy is for sure comparable to the human expert. At the same time, this comes with a classification speed up, right? If you have a trained model, this can actually happen from hours to seconds. It can happen anytime. It is more reproducible as human expert because you don't have any daily variations. So it comes with all the benefits. It also comes with a couple of downsides, of course. What we realized when doing that project was there's actually scarcity of data around. So when we had this classifier developed, we thought, okay, let's now go to real blood cells, human data, but there's not much around. The situation changed a bit, and we also tried to do our best here. So we published part of the code in something which is called Collaborators. You can go there and play with it. And we published the full data set together with the annotations in the Cancer Imaging Archive. And you can just go there, download it, and play with it. That was nice. And it also was one of the first applications of deep learning in cytology, which earned Christian Martek, at that time doctoral student in my lab, the Vision Zero Award 2021, just this year. Vision Zero here doesn't mean you don't see anything. It means that let's try to get rid of cancer altogether. So there was a high profile oncology symposium this year in Berlin. And actually, it even popped up in the Bild Zeitung. So Christian, congratulations to making it in that newspaper. I guess more media attention is hard to get for a scientist. Good. We can discriminate blast cells from benign leukocytes. Amazing. More relevant, though, is the question, can we classify blood diseases with AI, right? I mean, just identifying blood cells is nice, but it doesn't help any patient. So can we classify blood diseases with AI? For that, we need a different data set, right? So we now compile the data set where we have control, stem cell donors, and then 4 different AML subtypes. AML is, according to the WHO, now classified into a variety of subtypes, some of them genetically defined. And those 4 subtypes here are genetically defined, meaning we have a great ground truth. We know that this patient has a particular mutation, and this mutation leads to AML. This is a strong correlation with the Munich Leukemia Laboratory. Actually, they have this data. They now scan blood smears as a standard technique, and they also do the genetic test. So they have a great data set and shared it with us here for following up on that question. So what you basically have is roughly 200 patients. For each of the patients, between 200 to 400 of these single cell images. And if you look into one of those images from an AML PML RARA patient, you see actually in that particular cell our rods, which are these strange looking, very unusual morphologies inside the cell. So it's not the nucleus, but these things basically are diagnostic for that particular subtype. So it's a strong morphogenic correlation. At the same time, it has a diagnostic value, because if you see such our rods in the blood cells of a patient, you know that this is PML rara. And you know also that this needs very, very quick treatment, because only then the survival chances of this patient are high. So it's a needle in the haystack search, right? Finding that cell, identifying the rods in that cell. And that's, of course, something which we're interested in. Is AI able to do that job as well? Are we able to find the same cells? And is the algorithm actually concentrating on these cells as a human expert would do? Good, let's go for it. Problem is we don't have any single cell annotation in that case, right? In the last project, we had 80,000 leukocytes annotated. That's a very tedious process. It takes us to wait, to be patient for a couple of months, right, until we get the annotations back. Here, we didn't have any annotations, and we didn't try to get them. Instead, we have a bag label annotation, so not the instances that are labeled, but the bag. And the bag label is just the WHO classified disease, in that case, PML rara. So bag label available, instance label no. Luckily, there's a solution for that, which is called multiple instance learning. So machine learning has already developed a solution. We used that and we extended it with an attention mechanism, which allowed us to figure out how important a particular cell or instance is for the final classification. And you're going to see in a second how we apply that to our approach here, to our data set. So training that model, and then applying it to the 5-fold cross-validation on test sets, you see here that we can very nicely discriminate the stem cell donors from the MA patients. That's good. Anything else would be not very good, right? Because healthy people have to be clearly discriminated from MA patients. Interestingly, AML, PML rara, the particular disease I showed you a second ago, can also be nicely separated from the others. But also these are the 3 genetic subtypes where it's not totally clear if there's a strong morphogenic correlation. Also, they can be separated by the algorithm relatively well. Now, that's good. So we have the chance to discriminate these subdiseases. How are we doing that? So it's, for these health applications, it's super important to get an idea, an explanation how the algorithm actually does the decision making. So with our attention mechanism, which tells us again how important is the cell for the final prediction, we can actually look into that. So here you see all the single cells from one AML, PML rara patient, and we plotted the single cells here according to their attention. So high attention means important for classification of PML rara, low attention means not important, not used basically for the final classification. So we can plot them. Which is nice, but it doesn't tell you much again. So we went back to the MLL and asked an expert there to classify the cells now according to their importance for the classification of the disease. And if we now plot this label here, this is black for, this is a cell which is very suspicious for ML, versus white, where you say, no, this is no clinical, I can't say anything about the cells, and gray here, which means it's unsuspicious for ML, so that can be anything. You see that those cells with a high attention, right? These are the cells which are classified as suspicious for AML. So these are the cells where also the cytologist will look closer in order to find a diagnostic cell or to, put them in the class of a AML specific diagnostic cell. So very nice. Our algorithm looks at the same cells as the expert. This works for PML rara nicely. So here we show the most important cells for the disease. We can also do it for the others. For the stem cell donors, you see these are granulocytes, healthy cells. So nothing pops up here. But for the other cells, you also see interesting morphological patterns that come up. And we are going to look more into that to figure out if we find cell types here, which may be used as morphological indications later. What else can we do? We can take the features that we extract for each of the patches and embed them in a 2-dimensional landscape here, as we did using the UMAP algorithm. And if you then use the annotations from the expert, which I just showed you, on a few of the cells, right, not on the 200 patients, but just on 4 of them, you can basically get a very good idea about what these clusters mean, right? And for a single RNA-seq, you have to get the gene expression. Here you have 100 years of experience of what the cells actually how they look and what they are, right? So we can nicely annotate this map here, and we see now clusters for lymphoids, for eosinophils, myelocluster, and also you can follow the development of maturing granulocytes from myeloblasts over from myelocytes towards mature neutrophil granulocytes on the right-hand side in green. What can we use the map for? Well, we can just now project single patients into the map, which gives us a very quick view on what is peculiar about that patient. For example, PMRR, you see that we have here a lot of blasts which actually shouldn't show up in the blood. They should stay in the bone marrow. As soon as this bone marrow blood barrier is broken, they go into the blood and then pop up and are indicative of AML, of course. And also then you see for other AMLs, the pattern's a bit different, and we think that this might be a very nice way to assess very quickly how a patient is doing in terms of the different populations and also in terms of the frequencies of the cells. There's one more thing we can do, and this is something we call single cell occlusion. So let's take one of the cells out and figure out how that changes our final classification. By doing that, we can figure out if the absence or the presence of the cell is important for a final classification. Doing that for stem cell donors, you see that the presence of the healthy granulocytes on the right-hand side, this is important, right? But as well, the absence of all the blasts. So the algorithm uses both aspects in order to classify a patient into that class here. And this is interesting to look into the other four classes because it tells us how the algorithm actually decides depending on the presence or absence of particular cell types. So can we classify blood diseases with AI? I would say yes. This is to prove that we can at least discriminate these four AML subclasses, right? More relevant for patients is, of course, can we detect blood diseases with AI, right? Can we replace this tedious algorithmic inspection of single cells done in the lab with algorithms? And actually, to answer that question, we need a couple of other things, right? We need a realistic cohort in the first place, right? I mean, selecting 200 patients with well-defined AML diseases, that's one thing, but detecting PML-RARA in a realistic, let's say, input that the MLL, for example, has, that's a different question. We also need high-throughput scanning, right? Because we need all of these patients scanned to be able to analyze them. And we need robust models. So we need trained models that allow us to be applied, not just, let's say, 2020, but also 2025, right? When maybe the scanners have been changed a bit, or the resolution has been changed, or the lighting has changed. Or, of course, if you want to go from one lab to the other, where again, other parameters of this process might be changed. Senior robustness evaluation is one thing we looked a bit into. So what happens if you take one of those images and now you change it in terms of the brightness or the compression or the blur? And actually, Christian looked into that, and we just recently figured out that if you augment your training data, and you know the variation that you can expect, and you augment the training data, you can actually very nicely cope with these kind of variations and make your model more robust. And we look into other of these confounders at the moment. Right. How to understand blood stem cell kinetics is, of course, a very different question. And as I now told you the last 20 minutes about that part of the lab, I'm now going to talk about the mechanistic modeling part. Again, we know that this is our very rough abstracted view on hematopoiesis. Actually, we know much more about the process, both in mouse and human. As you see here on that picture, we have a very good understanding on how to identify the different subtypes, also a very good understanding what transitions are possible in this hierarchy. Still, there are a couple of open questions. For example, in a disease like AML, what is exactly happening there, right? I mean, we know that blasts accumulate in the bone marrow, but why? Is it only that they stop differentiating, or maybe are their rates regulated on the stem cell level or progenitor level? Are there maybe even new transitions in that hierarchy popping up? In order to look more into that, we teamed up with Katarina Götze at the Clinical Complex der ISA. Katarina has access to bone marrow samples from people that have a hip replacement, elderly people mostly, which is a great resource for healthy bone marrow. So we take these HSCs, Karina puts them into culture, and then what we just do is we count single cells over days. We look how these hematopoietic stem cells develop over time, how they differentiate, and we count these numbers. Now, we abstract this relatively complex hierarchy into something that we can model with a multi-compartment model. Here you see we have 7 different states. Each of the states has 3 possible reactions: division, cell death, differentiation. And with each of these reactions, we attach a rate, which gives us then a system of coupled ODE equations, what you have here. And if we now take this mathematical formulation of the process and we fit it to the data we have, you see that first we can nicely fit the data we see here. We also measured actually the number of divisions of the single cells, which gives us more information. You see that HSCs go down over time while the other cell types come up. And now, having that tool around, we can now ask a couple of questions. For example, if we look at the donor age of our cohort, is there something changing? And we find that HSC proliferation, though the number of divisions of the HSCs, actually this goes down with age, which is to be expected, right? The older you get, the less active your HSCs are probably. And we can also ask another question, and that is, there have been a couple of hierarchies actually proposed in literature with sometimes subtle changes, while A here is basically the standard model. We have a couple of other things proposed, often due to experiments or observations, also in challenged hematopoiesis. We wondered if we can now use the data from our cohort and figure out which of these models here fit best to the data we observe. And what we use for that is called the Bayesian information criterion. It's a model selection tool. Basically, we look at the likelihood of an optimized model, how likely or how well does the data fit to the model predictions. Basically, we have the parameters optimized for each of the model, and then we penalize each model for the number of parameters. So more complex models get a penalty here, which allows us to compare the different models. And doing that for a healthy cohort, we found that we can reject 7 of these proposed models. They definitely fit not to the data we observe in our healthy cohort, but 3 models remain here, A, B, and C. So these are basically possible and plausible from our modeling point of view. Now, recently, Katarina published a paper where she shows that the bone marrow from these hip replacement people, actually, they are not all healthy. There is something called CHIP. This is clonal hematopoiesis of, what is it again? CHIP of clonal hematopoiesis of, somebody may help me out here. Let's look it up in a second. It's basically a pre-form of AML and also another disease which is called MDS. People are not sure if this clonal hematopoiesis means anything. Clonal hematopoiesis disrupts the fact that from these many stem cells you have in the bone marrow in the beginning, more and more die over time, and you are left basically with only few stem cells in your bone marrow. They produce all the blood, so you have a clonal production of blood, and you can observe that. And the question is, does it have any effect on further disease development, but also on the kinetics of your blood production? So you have to take these healthy cohorts from hip replacements with a bit of caution, and we checked actually what happens to our cohort, and indeed we find that again 50% roughly of our healthy people are CHIP patients. Here on the left-hand side, they have a particular mutation which is then popping up in all your blood cells. This is our cohort here on the left-hand side. On the right-hand side, this is a cohort of MDS patients, multiple dysplastic syndrome. This is a pre-form of AML, again signified by a couple of mutations in your blood cells, and people suffering from MDS show already symptoms like reduced energy and again a high risk to develop AML. Now, we took that same cohort, put it in our model, and figured out which of the rates in a combined model of these A, B, and G models I showed you before are actually changed. And interestingly, find some rates are changed in the CHIP patients already. So we have a higher differentiation rate here, for example, in 2 out of these 5 patients. If you look at MDS, again, we have higher differentiation rates, also more than CHIP, but still the pattern you observe here is very heterogeneous. So there's not one rate which is changed overall in all of the patients. It's a heterogeneous picture, but that actually fits also to the genetic heterogeneity of the cohort, which I showed you before, which is also a hallmark of that syndrome here. Right, I showed you two aspects of what the lab is doing on hematopoiesis, improving image-based diagnostics with deep learning. On the one hand, predicting single-cell kinetics, understanding processes during hematopoiesis with mechanistic models. And of course, in the future, we would love to merge these things closer together by using the data we extract from the blood smears and the bone marrow smears, for example, to parameterize our models. And on the other hand, using that models to constrain the deep learning rates and architectures. We're going to open up a new institute at the Helmer Center, which is called AI for Health. I'm very much looking forward to that, also to getting two colleagues on the senior director side. So if you're looking for a job in Munich, consider applying for a senior directorship position at the Institute of AI for Health. In my lab, we're going to continue work on what we do here, develop models for clinical needs, merging, as I just said, mechanistic and statistical models, and also trying to close the proof concept to production gap, because what I showed you here, of course, that's research, right? We try to get these things into the clinics and into the labs to make a difference for patient life. Many thanks to our collaborators, right? Everything I showed you is very much application and clinical question driven. So without these great collaborators, it wouldn't be possible. Thanks also to the other people which I collaborate with, which I didn't talk too much about. Also thanks to Fabian, Anna, and Alf, who's providing the computational infrastructure at the Helmer Center for our work. As I said, the Marr lab also works on other projects which I didn't touch on. Here is this amazing bunch of motivated young people I have the pleasure to work with. If you consider coming here, please do so. There are a couple of vacancies also for PhDs and postdocs. So if you in particular think about merging deep learning with mechanistic models, please write me an email and let me know. Thanks for the funding, buddies, and thank you very much for your attention. Looking forward to questions and the weekend.
Well, thank you, Carsten. That was highly inspirational and very cool work. Thank you. So we have a couple of questions in the chat, and as a reminder, you can also raise your hand to ask questions yourself. One question is, where do you see those approaches going in terms of a clinical applications? So the precise question is, what do you see AI moving from diagnostics in hematopoiesis to prognosis of the disease course of patients? So what are the major hindrances and roadblocks that you expect towards clinical applications as well?
Yeah. There are a couple of interesting works on that, right? Predicting tumor grades, for example, from histopathological sections, right? There's a lot of work being done at the moment. The question is, I guess, indeed, when are we going to be able to use that for, daily work, for really patient life? And there, I guess, the hindrances are, as I said, right, robust models that do not work only on a particular cohort, but on any type of data you put in. Also, there are a couple of interesting questions, approaches are developed. Approval, of course, of these methods from the regulatory bodies, and then getting the stuff really into a workflow that also allows clinicians to use them efficiently. So I guess we are on a good path there, but it will take a couple of years, I guess, until we can really use it on a daily basis in the clinics.
Mm-hmm. So I had a follow-up question to this, which is basically how reflective are your training data of what's really going on in the population? Do you have any sense of sensing that?
Yeah, so what I showed you is a particular cohort out of the data set from the Munich leukemia laboratory, right? So this laboratory is one of the largest in Europe. So their input is relatively representative of what is really used in order to make predictions for patients, right? So they have this cohort. Still, I mean, what I showed you, right, is a very selected cohort. We are now trying exactly that. So trying to figure out how well is our model able to perform on a realistic cohort where you don't only have healthy patients AML, but you have any type of diseases in there, right? So out of bag prediction here, of figuring out this is AML, this is something different. These are the challenges we are faced here at the moment, and this is exactly what we are going towards in this collaboration with MLL.
Mm-hmm. Then we have a question from Deso Modus. Hi Carsten, this is a really nice talk. Different labs, how robust were the models?
Very good question. We have one paper coming out in Blood very soon where the reviewers asked us exactly that. There wasn't blood, but bone marrow smears. Whenever you try to assess that, it's becoming a bit tricky because so what we did there, we had a nice curated data set from our lab basically together with colleagues from Erlangen and the MLL again. Trained a model, evaluated on the data set, right? Then we tried to figure out how well that works on a different data set. So we looked around again, as I told you, there is not too much data around, right? So people are very holding back on the data set. Again, I ask everybody to publish the data and in a cleaned up form. So we eventually found a smaller data set from a Japanese group, and we applied our model on that. What we saw there is actually, it's pretty hard to quantify, right? Because they have a different annotation scheme in the first place. I mean, we use 20 different classes, they have 11. And then if you talk to morphologists or cytologists, right, even how they define the classes is different, right? So that's a different school of how you define cells in Germany as compared to Switzerland, as compared to Japan, right? So what we try to do is forming meta classes basically, right, that allowed us to quantify the effect. What we saw there is at least that our model is able to perform relatively robustly, so we can identify or we can discriminate different of these meta cell classes, but a detailed analysis would require that you take the same data set and ask again an annotator in Japan and here in order to classify the cells. So we need more annotations on these data sets in order to be able to really quantify how robust the models are. Still, you're totally right. That's the next step, right? Making the models more robust, also to perform on different data sets. Although this depends on the question, right? I mean, for the MLL, for example, I mean, they don't need a model that works on Japanese data, right? They have their input. They want to have a model that works robustly on their input, and that's enough.
Mm-hmm. Thanks. Then a question from Anne-Rigor. Could you go back to the basics of the occlusion maps? Could you revisit those?
On my slides?
Yeah.
Oh.
And explain a bit more the intuition behind them.
Right, let me try to show you these images. Here we go. You still see my screen, right?
We do.
Yeah. So we have roughly 100,000 of these single cell images. So we can just take now, so and for each patient, right, we have a couple of hundred of those. So if we leave out one of the cells for the prediction, we can check how the prediction probability for the 5 different classes changes, right? So taking out a cell lets us know if this cell actually enhances or reduces the score for the particular disease. If you do it for the cells, you can produce these cell maps, which indicate you then which cells where in the map are important in terms of their absence or presence for the final classification, which is nice because actually we don't need single cell annotation here, right? We know from the annotation of a few cells what the map actually, or what the different regions in the map display. So it gives us an idea which kind of cell types are actually responsible for the final classification of the cell type. If you look closely, I guess these maps are still a bit rough, and I hope if we double or triple our data set, this will get more smooth, because I guess a couple of these highlighted cells here are maybe still in the artifact class, right, where a particular cell from one particular patient now dominates that score here.
Mm-hmm. Thanks. So I'm interested in what do your models actually learn? So thinking a bit into the direction of generative models, can you actually predict what an AML cell looks like just based on what you learned from the models?
That's a very nice question, Fabian. We're looking into that. We work on autoencoders, which would allow us to also then basically predict how they look. One nice idea I would love to have at some point is a movie of how cells differentiate from all the data we have here. We are not there yet, but that's definitely something we should be able to do given the vast amount of data that is around now.
Mm-hmm. Thanks. Let's check if we have more questions. So there's one student interested in master and PhD positions. So I suggest he or she gets into contact directly with you.
Write me a mail, send me CV, your transcripts, and why you want to come to us, as usual.
Okay, great. Then I don't see any more questions from the chat.
I have something to say. CHIP stands for clonal hematopoiesis of indeterminate potential. Sorry for missing out on that.
Oh, okay, yeah. I didn't see a question there, but okay. Care to elaborate on this?
No, I just during the talk I couldn't recall what the IP stands for. So indeterminate potential.
All right. Well, then thanks for the participation from the chat, and thank you so much for this great talk. We see each other again in September. I wish everyone a great summer. Enjoy the summer.
Thanks, everybody. Take care. Ciao.